Good morning, afternoon, or evening, wherever you are, everyone. My name is Diya Pendyala, and today I will be presenting on the future of preventative medicine, in other terms, CRISPR. The topic that I researched these past few weeks was a comprehensive analysis detailing the processes of CRISPRi and CRISPRa inhibiting and activating specific genes using pooled and sensitization screens and their implementation in curative medicine. I worked with an amazing PhD student named Jacob Odell, who mentored me throughout the research process and helped me reach my results. I would like to thank him for his support and his patience in answering all of my questions. Now, I will be talking a little bit about the background behind my findings. What exactly is CRISPR? The full form of CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it is a system currently used to cleave and either silence or activate certain sections in an organism's DNA. It was first discovered in bacteria immune systems, and as you can see in the diagram, when viral enemies attack specific bacteria species, a snippet of viral spacer DNA would be inserted into the bacteria sequence. From this, a CRISPR RNA or a cRNA sequence would be produced, and this sequence would be inserted into the viral enemy's body. This would deactivate or kill the enemy and save the bacteria from more attacks. Once this was discovered, many scientists, including the famous Jennifer Doudna and her team, performed many in vitro experiments to determine the exact components of CRISPR RNA strands used to cleave and edit DNA strands, so cutting the DNA strands in half or editing them. Throughout the bulk of these experiments, they invented and tested the use of chimeric RNA strands, which means that the strand contained a CRISPR RNA or a cRNA and a tracer RNA sequence. So these single-stranded RNA sequences, the chimeric RNAs, were the revolution of gene editing. These proved to be able to target any gene in question and cleave it at a specific location, tailored and targeted with this RNA sequence. And this opened up all possibilities for gene editing. So this was the revolutionary start. Going over the methods and analysis that I used for this project. So the main methodology that I used was case study. So for my research paper specifically, many articles were used to gain a better understanding of the history of CRISPR. So again, the experiments that were carried out by Jennifer Doudna and her team, the biochemistry behind it, again, talking about the CRISPR, the tracer RNAs, and the chimeric RNAs, and the ways that this technology can be used in many different applications, like CRISPR, CRISPRa and I, and RNAi. We also used 3 or 4 possible case studies to analyze and determine a logical opinion on the ethics behind CRISPR using germline and somatic cell editing, which I will explain later, in humans in the near future. And analysis and opinions were included in my research paper, as well as information for everyone to understand. Finally, I will be going over a summary of all my findings. On the right side of the slide, you can see the names of different CRISPR technologies that are currently undergoing trial and research. CRISPR, the regular version, the one in red, is unique in its own way because it edits the DNA at the DNA level itself, so physically editing the nucleotides A, C, T, and G. And this makes it more permanent when edited this way, it edits itself. CRISPRa or I, which is the one in yellow, stands for CRISPR activation or inhibition, which means that the technologies edit at the RNA transcription level or the protein production itself. These edits are less permanent and affect the actual activation or the inhibition of the gene instead of the gene itself, unlike in regular CRISPR. RNAi is quite similar to CRISPRi and A. However, it can only inhibit gene expression, not activate. So it cannot do what CRISPRa can do. This makes it quite limited in its abilities. Next, I'll be talking about genetic screening. Genetic screening is a process used for selective specification of genes or choosing genes for mechanical follow-up experiments. Depending on the method used, such as CRISPR or CRISPRa and I, different screening methods are used, including pooled or sensitization screens. Lastly, I'll be discussing the ethics behind both somatic and germline editing. So the future of CRISPR is to be used in later generations to edit the DNA of humans themselves. So far, we've been looking for disease-related edits, so stopping sickle cell anemia, cystic fibrosis, other single gene disorders that are controlled by genes, which means we can edit them and stop them from happening. However, there's four types of editing in total. You have somatic versus germline. Somatic means you're editing only body cell genes, so it only affects the host organism, it doesn't affect later generations. Or germline cell editing, meaning you're editing on the sex cells, which means that your edits will carry out throughout generations. You also have cosmetic edits, which means that you're editing the color of your eyes, the color of your hair, things that you might want to change for your later generations, or again, disease related, like we're looking forward to. And so it's nice to think about these ethical implications. So if you're editing your child or you're editing future generations, is this something your child would have wanted? Is this something that they would be okay with, considering the social standards and the social impacts of humanity? You also might want to consider cosmetic versus disease things. So like, editing your child's eye color or hair color, is this what they want? Maybe that you're not considering exactly what the unborn child wants, versus using just disease edits might be something that's obviously objectively for the child's good. I would like to thank you for listening and urge you to think about the future of this technology. These editing technologies are expected to go through human trials for inhibiting certain diseases, including sickle cell anemia, very soon. Are you okay with this technology, or do you have certain reservations on how it can be used? Thank you for listening.